Good morning. It's August 26th, and this is your daily brief in science. Here's everything you need to know. Eisai-and-biogen have launched leqembi, also known as lecanemab, in Austria and Germany, marking the first European Union countries to approve this Alzheimer's treatment. The drug is an anti-amyloid beta monoclonal antibody that has shown a 31% reduction in clinical decline over 18 months in clinical trials. This indicates potential in slowing the progression of early Alzheimer's disease. Common adverse reactions include infusion-related reactions, headaches, and ARIA, with serious ARIA-E cases occurring in about 2% of participants. The launch aims to improve access to advanced treatments for early-stage Alzheimer's in Europe, addressing a significant medical need. Eisai leads global development and regulatory efforts for lecanemab, with both companies co-promoting it in the European Union, where Eisai is the marketing authorization holder. The companies have a long-standing collaboration, with Eisai responsible for development and commercialization and BioArctic holding rights to the Nordic region. The launch involved working with healthcare authorities and implementing controlled access programs to ensure safe use, especially considering safety concerns like ARIA. Lecanemab has been approved in 48 countries, with ongoing reviews elsewhere, including recent U.S. approvals for additional dosing methods such as intravenous and subcutaneous autoinjectors. Further studies are underway, including trials in preclinical Alzheimer's and for inherited forms, exploring early intervention and broader applications. The European Union launch highlights the importance of early intervention in Alzheimer's, supported by clinical trial data and regulatory pathways. Overall, this development represents a significant step forward in addressing unmet needs in Alzheimer's treatment. A recent breakthrough in transplant science involves a lung xenotransplantation using a gene-edited pig's lung on a 39-year-old brain-dead patient. This procedure, performed in China and published in Nature Medicine, represents a significant step toward addressing the global shortage of donor organs. The transplanted lung remained functional for 9 days, despite evidence of immune response and tissue damage. This experiment builds on previous efforts with pig hearts, kidneys, and livers, but lungs present unique challenges due to their exposure to environmental threats and higher rejection risks. The study highlights both progress and ongoing obstacles in xenotransplantation, including immune reactions and the presence of native lung tissue. Experts note that this research is still in the experimental phase, with further studies needed to evaluate long-term viability and safety. Currently, lung transplants face long wait times and limited supply, making pig organs a potential alternative. Future research plans include more transplants into brain-dead patients, testing bilateral procedures, and refining genetic and immunosuppressive techniques. While successes with pig kidneys and hearts are promising, significant hurdles remain before xenotransplantation can become routine. Key challenges involve managing immune responses, improving genetic modifications, and ensuring long-term organ function. Additional research is exploring growing humanized organs inside pigs and restoring human donor lungs to usable conditions. Ethical considerations remain prominent, including concerns about animal welfare, patient consent, and the possibility of creating a two-tiered system where some patients receive animal organs instead of human ones. Recent research utilizing apoplastic metabolomics has identified sugars such as sucrose, fructose, glucose, and sorbose as key metabolites that promote stomatal opening in response to red light in plants like Vicia faba and Arabidopsis thaliana. This study uncovers the molecular signaling mechanisms controlling stomatal movement, showing how sugars activate pathways in guard cells that regulate ion fluxes, particularly potassium and chloride ions. These ion movements influence guard cell turgor and aperture, linking metabolic activity to gas exchange. At the cellular level, sugars play a crucial role in connecting photosynthetic activity with environmental cues, forming a feedback loop. The findings suggest the apoplast is an active signaling space that integrates light perception with cellular communication, shifting traditional views of sugar functions. Understanding these internal plant communication pathways could inform future strategies to improve crop resilience and yields under stress conditions like drought and heat. The research highlights how plants optimize carbon gain while minimizing water loss, providing insights into adaptation mechanisms. These discoveries have implications for agriculture, indicating that manipulating sugar signaling pathways may enhance crop resilience and water use efficiency. Funded by the U.S. National Science Foundation and involving collaborations between U.S. and Japanese institutions, the study emphasizes the importance of integrated metabolomic and physiological approaches. It expands the traditional view of sugars as merely energy sources, emphasizing their role as dynamic signaling agents. Future research may explore specific sugar types, their transport mechanisms through the apoplast, and interactions with other guard cell signaling pathways, such as abscisic acid and calcium signaling. Experiments show that sucrose enhances stomatal opening independently of guard cell photosynthesis and does not require phytochrome B signaling, confirmed by studies involving photosystem II inhibitors. Researchers have identified high levels of a molecule called chorosin in the blood of patients with diabetic kidney fibrosis, a condition that worsens kidney function. Chorosin is produced by gut bacteria such as Staphylococcus species and can travel from the intestine to the kidneys by attaching to albumin in the bloodstream. Once attached, chorosin detaches in the kidneys, where it damages kidney tissues and accelerates disease progression. The molecule interacts with albumin, which helps it move through the blood and increases its stability and damaging activity. Chorosin contributes to kidney damage by promoting aging, inflammation, cell death, and scarring, all of which are features of diabetic chronic kidney disease. The study, published in Nature Communications, integrates microbiology, immunology, and clinical sciences, and suggests that targeting chorosin could lead to new therapies for CKD. Laboratory experiments and molecular simulations show that chorosin reaches the kidneys by attaching to albumin, which acts as a carrier. Future research plans include testing anti-chorosin therapies in larger animal models like pigs to assess safety and effectiveness. These studies aim to validate chorosin's role in kidney fibrosis and develop treatments for humans. Chorosin's interaction with albumin not only stabilizes the molecule, but also amplifies its harmful effects in diabetic nephropathy. The primary source of chorosin in diabetic patients with CKD is identified as Staphylococcus species, with bacterial peptides found in urine samples. Chorosin bypasses immune detection by attaching to albumin, then detaching in the kidneys to trigger damaging inflammatory pathways. This interdisciplinary research opens new possibilities for microbiome-based therapies to treat chronic kidney disease. Researchers have developed Decipher, an AI-assisted tool that identifies how tuberculosis drugs kill bacteria at the molecular level. This technology can distinguish whether a drug targets the bacterial cell wall or disrupts energy production, providing detailed insights into drug mechanisms. In a recent study published in Cell Systems, Decipher revealed that a clinical TB drug impairs the bacteria's respiratory chain rather than destroying its cell wall, challenging previous assumptions. This advancement allows scientists to evaluate various drug combinations and conditions more rapidly, which could accelerate the development of more effective treatments. TB remains a major infectious disease that requires long-term multi-drug therapy, with increasing drug resistance complicating treatment efforts. The new method offers a cost-effective and faster alternative to traditional techniques, linking bacterial structural changes to gene expression profiles. The research team plans to validate these findings and integrate Decipher into clinical practice to speed up the creation of new therapies. They also aim to foster global collaborations to enhance TB drug development and explore applications for other infectious diseases and cancer. Beyond TB, Decipher's technology could support the development of treatments for various diseases, improving drug discovery processes worldwide. The approach reduces costs and time, making advanced drug testing more accessible, especially in resource-limited settings where TB is most prevalent. This has been your Daily Brief in Science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.